Hello, this is Dr. Michael Edwards, founder and CEO of Bioinfo Solutions, a data analytics company based out of Parker, Colorado. This entry is part 5 in a series of instructional videos detailing a meta-analysis on 8 different human gene expression studies looking at papillary renal cell carcinoma. In this installment, I use Partek Genomics Suite to select a group of clustered genes in our meta-analysis. I then import these into the networking platform String, which is a freeware gene-protein interaction network, and Elsevier's Pathway Studio, which is a commercial product, to derive biological meaning from our list of genes. A text file containing a list of the clustered genes that I use in this video, along with the normalized fold chain scores for each comparison, can be downloaded at a Google link provided in the description of this YouTube entry. When we last left off on our video series, we had filtered our original 5,000 genes in our meta-analysis down to 2,000, which comprises those top 1,000 genes with the highest sum of normalized scores across all the comparisons, and the bottom 1,000 with the lowest fold changes, the sum of fold changes across all 17 different comparisons. Again, this is just a way for us to not bias our data. I want to look for things that go up and down. We also formatted our heat map to include the control tissue, the type of test cancer that we were comparing against our control, and then the subtype of that cancer. Right now we've got, and again we clustered based on Spearman dissimilarity for both the comparisons and the genes. Just so we can see the data a little better, what I want to do is get rid of this. So I'm going to go to my dendrogram and I'm going to say do not show this dendrogram and hit apply. Now that we have these data in this visual format, we can start to see trends in the different comparisons across all the different studies we looked at. Blue, again, blue, the more intense the blue color, the more a decrease in expression of that gene compared to the control, which is given in our top bar. The more intense the yellow color, the higher the expression was compared to that control, and black would be that we did not find a significant difference in expression based on the previous criteria that I've given you. What amazes me is how consistent these data are. That if a gene went down in, say, type 1, it tended to also go down in type 2 compared to the control. And again, the majority of these comparisons over here are cancer versus normal tissue. And you can see they're very consistent, fairly consistent across the different comparisons. Although what we're going to do is we're going to exploit some of these differences here where we're not maybe seeing a change in type 1, but we are seeing a change in type 2. We're also going to use these last two columns here to kind of guide which clusters that we pick. Again, these last two columns were cancer versus cancer. So type 2, the more severe form of papillary renal cell carcinoma versus type 1, and poor versus excellent survival of papillary renal cell carcinoma. And as you can see here, they are very, very similar, and they were done by two different groups. So type 2 and type 1 and poor survival versus excellent survival look very, very similar. So these are what I'm going to find are going to be some of the interesting clusters that we're going to pull out. What are the differences between these two different types of cancers? Can we characterize the different forms of cancers based on the patterns of expressions that we're clustering here? And as you can see, and what I find fascinating, is you can see that you have these group of genes that go up in type 2 versus normal. And again, you're seeing here that not very much of a change when compared to in your type 1 versus normal. And then when we look at the direct comparisons of type 2 versus type 1, or even poor versus excellent survival, we are seeing also an increase as well. So what I want to do is I kind of want to get to this cluster. I want to see what are the differences here. What are these genes that are increased in the more severe forms of this cancer? What does it have to say about that cancer? What are these genes? What are some of the biological processes that are involved in? So in order to do that, we can just select some of these clusters, and I'm going to do that right here. I think I can get to it. So what I find interesting is this particular point here, where you're seeing that there's not a lot of expression or much change between our type 1 versus our normal tissue, but we are seeing a significant increase in the type 2 versus the normal. And again, we do see that difference in the cancer versus cancer comparisons. So in order to select this cluster, what I'm going to do is just draw a box and I select it there. If we want to get a closer look at some of these genes we selected, we just scroll in here. If we scroll in far enough, we can actually see the gene symbols for those particular genes. One of those being aurora kinase A. I see that a lot, and it upregulated in a lot of other different types of really nasty cancers. We can also scroll out a bit here to see the pattern of the expression or the expression patterns of the genes we selected. Again, these are given in here. Let me scroll out a little bit more. So now we can see, again, the genes that we selected tend to, in the type 1 or lower grade forms of papillary renal cell carcinoma, tend to either not be expressed or not expressed as highly as the type 2 versus the normal kidney tissue. And you can see that in these three columns, the expression is very, very high, and which is also confirmed in our last two columns, again, which is comparing the more severe types to the less severe forms of the cancer. So again, we're getting confirmation not only with the cancer versus normal tissue, but also the cancer versus cancer. So these are the genes I want to select. And it is my theory, given the fact that these fold changes, these scale fold changes, seem to be similar over all these different comparisons for these particular genes, and that they seem to be more active in the more severe forms, or the type 2 versus the type 1, I think that they are acting together, that they are enriched for biological units, pathways, connections. And so what I want to do is pull these genes out and put them into another program where we can actually, you know, put the network together. So in order to do that, all I have to do is put my cursor over this and right click. I go dump selected to spreadsheet. I want to keep it in the original heat map order, so it'll give me my genes in the rows, and I hit OK. And as I do that, there should be a list right here of the genes I selected, and again, these are 130 genes again that were higher in the more severe forms of this cancer compared to the type 1 or the less severe forms. So in order to now what we're going to do is take these genes and use another program, which is string, to pull out the biological meaning from this list, number one, and number two, to find, how are these things connected? Where are these relationships? And is there, say, a central molecule that we can possibly see that might control much of the gene expression we're seeing here? So in order to do that, I'm just going to put my cursor on the top cell here, go all the way to the bottom, hold down my shift key, hit select, go up here to edit, and I copy that. And now I am going to paste this onto paste this into string and now pull out the biology. So that's what we'll do in the next series of videos, is I will take these 130 genes and try to figure out not only how they're connected, but maybe some particular pathways or biological functions they're associated with.